When you look at a skyscraper, you see the glass, the steel, the sweeping architecture. You register the whole building as just one single cohesive entity.
Right.
You don't usually think about the thousands of individual nuts and bolts and like the microscopic welds holding all that internal scaffolding together. That is, until one of those bolts snaps.
Exactly. And in structural engineering, that invisible microscopic failure cascades. The stress shifts to other joints, materials begin to warp, and eventually, well, the entire system is just compromised.
Yeah, and that structural reality, it maps perfectly onto the human body. We look in the mirror and we just see a person, right? A single entity. But underneath, we are this staggering, highly regulated chemical reaction.
We really are.
So today, we are pulling from a foundational text, Oraby's Illustrated Reviews of Biochemistry, to really examine those invisible nuts and bolts that are keeping you alive right now. Our mission for this deep dive is to take these incredibly complex molecular structures and translate them into the practical realities of health and disease.
Which is so important for anyone studying medicine or really just curious about their own biology.
Okay, let's unpack this. We're going to journey from the basic raw materials your body is built from, right up to the specialized machinery that runs the construction site, how those materials cross cellular borders, and finally, what happens when that whole biochemical supply chain just collapses.
And the true beauty of biochemistry lies in those mechanisms. It's not just a catalog of molecules to memorize. It is the study of causality. Understanding the precise reasons why, say, a single missing atom can lead to a systemic disease, it changes how you view medicine entirely.
It totally does. But we have to start with the raw materials. If we're building that biological skyscraper, we're looking at carbohydrates, lipids, and proteins. And going through Oraby's text, it becomes super clear that we vastly oversimplify these in our daily lives. Take carbohydrates, for example.
Right. The common perception is that they're just fuel, sugar rushes or carb loading before a marathon. But they do provide energy, yes. But their structural and regulatory roles are far more intricate than that. The source details a group of complex carbohydrates called glycosaminoglycans, or GAGs. A prime example here is heparin.
Wait, heparin? Like the drug?
Yes, but heparin is actually a naturally occurring carbohydrate found inside mast cells, which line our blood vessels, and it acts as a crucial anticoagulant.
Okay, I am struggling to understand how a sugar molecule functions as a blood thinner. Like, how does that work?
Well, it really comes down to physical shape and electrical charge. Heparin molecules are highly negatively charged. So when they're released into the bloodstream, they bind to a specific protein called antithrombin III.
Okay.
And this binding physically changes the shape of the antithrombin. It hyperactivates it so it can just rapidly neutralize the enzymes that are responsible for blood clotting. So the carbohydrate isn't acting as fuel at all, it's acting as a molecular switch.
A switch to maintain fluid dynamics in your circulatory system.
Exactly.
That completely flips how I usually think about carbs. And we see a really similar dual nature when we look at lipids or fats. I mean, there are triglycerides, which are basically the body's long-term energy vaults. But then there are phospholipids, which seem entirely focused on cellular architecture.
Yeah, phospholipids are the perfect building material because they are amphipathic, meaning they possess a hydrophilic or water-loving head and a hydrophobic, water-repelling tail.
They kind of have a split personality.
Right, and this physical property forces them to automatically arrange themselves into these bilayer sheets whenever they're in a watery environment. That creates the foundational membrane of literally every cell in your body.
Wow.
But Oraby's text highlights a very specific, specialized phospholipid called dipalmitoyl lecithin.
Oh, the lung surfactant.
Yes. This lipid is continuously secreted by the cells in your alveoli. Those are the microscopic air sacs in your lungs. And inside those sacs, there's a very thin layer of water. Now, water molecules are highly attracted to each other, which creates surface tension.
Right, they want to beat up.
Exactly. So if left unchecked, that tension would cause the delicate walls of those air sacs to just collapse and stick together every single time you exhale.
Oh, man. Like trying to pull apart two pieces of wet plastic wrap.
That is a very apt visual. So dipalmitoyl lecithin inserts itself between those water molecules, which drastically lowers the surface tension and keeps the alveoli open. And this mechanism is absolutely critical in neonatology.
Because of premature babies, right?
Yes. Premature infants often just lack the cellular maturity to secrete enough of this surfactant. The result is respiratory distress syndrome. The lungs literally collapse under the physical force of their own surface tension.
That's terrifying.
It is. But modern medicine combats this by placing the infant in an incubator and actually administering synthetic surfactant directly into the airways, physically propping those microscopic sacs open.
It's just sobering to realize that a newborn's first breath relies entirely on the successful synthesis of one specific lipid.
It really puts it into perspective.
Which brings us to the third material, proteins. These are the workers in the physical scaffolding. The source explains how amino acids link together via peptide bonds to form just a simple chain, right? The primary structure.
Correct.
Then they fold into spirals and sheets, and then they fold again into these massive, complex 3D structures. I often picture it like taking a flat piece of paper and folding an incredibly intricate 3D origami crane.
I like that analogy because the function of any protein is entirely dependent on that final three-dimensional geometry. Consider hemoglobin, which has a quaternary structure, meaning it's 4 separate folded protein chains.
2 alpha and 2 beta, right?
Exactly. 2 alpha, 2 beta, interlocked to function as one single unit, and its sole purpose is to transport oxygen.
And the text points out that this complex machinery can be completely broken by the smallest possible error.
It's just a single amino acid substitution. In the beta chain of hemoglobin, the 6th position is normally occupied by glutamate. Now, glutamate is hydrophilic, so it interacts really well with the watery environment of the blood. But in sickle cell anemia, a genetic mutation swaps that glutamate for valine.
And valine is hydrophobic.
Yes.
Here's where it gets really interesting. Because valine hates water, the protein suddenly has this massive mechanical flaw.
Right. It creates a sticky hydrophobic patch on the outside of the hemoglobin molecule. Under low oxygen conditions, these hemoglobin molecules actually try to hide their hydrophobic valine patches from the water by sticking to each other.
Oh, wow.
So they clump together, forming these long, rigid fibrous chains inside the red blood cell. And these physical fibers distort the normally flexible donut-shaped red blood cell into a rigid sickle shape.
Which then causes all sorts of problems.
Exactly. The cells get trapped in small capillaries, blocking blood flow, causing severe pain and tissue damage. It's a perfect example of how structure dictates function.
But wait, if that folded 3D structure is so delicate, what happens when it encounters extreme environments? Like if I take my intricate origami crane and drop it into a puddle, it's completely ruined.
Well, actually, dropping the paper crane in a puddle destroys the paper itself. The material dissolves. In biochemistry, that would be hydrolysis or digestion, where the actual peptide bonds between the amino acids are broken.
Oh, I see.
What you are thinking of is denaturation. Denaturation is more like taking that folded crane and carefully pulling it back apart into a flat, useless square of paper.
Ah. So the raw material is still perfectly intact, but it's just lost its geometry.
Correct. When you introduce extreme heat or strong acids, the weak hydrogen bonds holding the 3D shape together just snap. The protein unwinds. It loses its biological function. But interestingly, this is highly beneficial for human nutrition.
By cooking.
Yes. When we apply heat to meat, we denature the tough structural protein called collagen.
Turning it into gelatin.
Exactly. The tight triple helix of collagen unwinds. It can no longer support muscle tissue, obviously, but that unwound gelatin is highly soluble and incredibly easy for our digestive enzymes to break down and absorb.
Okay, so we have our steel, our glass, and our scaffolding. But raw materials don't just build themselves. The body needs machinery to assemble, dismantle, and regulate the site. That introduces enzymes.
Yes, enzymes are specialized protein catalysts. Their entire job is to lower the activation energy required for a chemical reaction. They grab molecules, orient them perfectly, and force them to react.
Speeding things up.
Speeding biological processes up by millions of times. And the best part is they emerge from the reaction completely unchanged, ready to work again.
But different tissues run different operations, so they need slightly different machinery, right? The source dives into isoenzymes, which I found clinically fascinating. Uses lactate dehydrogenase, or LDH, as an example.
Right, so isoenzymes are basically variations of the same enzyme. They catalyze the exact same chemical reaction, but they are physically structured to operate in different environments. So the heart primarily relies on aerobic metabolism using oxygen, so it uses the isoenzyme LDH1, which is optimized for that specific environment.
Okay, and the other tissues?
Well, the liver and skeletal muscles handle anaerobic environments better, so they utilize LDH5.
But how does a hospital lab actually tell them apart if they do the exact same job?
It comes down to physical chemistry. Because they're built from slightly different sequences of amino acids, LDH1 and LDH5 have different overall electrical charges.
Ah, I see where this is going.
Yeah, so if a patient arrives with chest pain, the lab places a blood serum sample in an electrical field, a process called electrophoresis. The isoenzymes migrate at different speeds based on their charge. If the lab sees a massive spike in LDH1, it means heart muscle cells have ruptured and leaked their specific machinery into the blood.
Wow.
It's a definitive diagnostic marker for a myocardial infarction or heart attack.
It is essentially a chemical fingerprint left at the scene of the crime.
Mm.
But we also have to regulate these enzymes, right? We can't have the machinery running at maximum speed constantly.
No, we regulate them using inhibitors. And there are multiple types, but competitive inhibition is the easiest to visualize, and it's the most heavily utilized in pharmacology.
Okay.
In competitive inhibition, a drug molecule is designed to look structurally identical to the enzyme's normal target, or substrate.
So if the enzyme is the lock and the normal molecule is the key, the drug is basically a perfectly carved fake key. It slides into the lock, but it won't actually turn. And while it's jammed in there, the real key is just blocked.
That is a perfect physical representation, and what's fascinating here is how we use this in medicine. Take bacterial infections, for instance. Bacteria need a substance called PABA to synthesize folic acid, which they require for DNA replication.
Okay, so how do we stop?
The antibiotic sulfanilamide is our fake key. It looks so structurally similar to PABA that the bacterial enzyme binds to the drug instead. The enzyme gets stuck, folic acid synthesis grinds to a halt, and the bacteria simply die from an inability to replicate.
We are literally starving them at the molecular level.
And wait, the source mentioned anticoagulants too, right?
Yes, dicumarol and warfarin. They are competitive inhibitors of vitamin K. By blocking the enzymes that rely on vitamin K, they stop blood clotting, which makes them powerful medical anticoagulants.
Okay, so drugs interact with enzymes, but vitamins play a massive role here too, acting as coenzymes.
Absolutely. Without a coenzyme, the main protein portion of the enzyme, which we call the apoenzyme, is functionally blind and inactive. The vitamin actually docks into the enzyme and completes the active site.
Got it.
For example, vitamin C, or ascorbic acid, is required for the hydroxylation of proline and lysine amino acids.
Which goes right back to the collagen we discussed earlier. Exactly. Hydroxylation adds an oxygen-hydrogen group to those amino acids. These groups act like molecular grappling hooks, allowing the collagen strands to cross-link and form a tough, durable lattice.
So what happens if you don't have vitamin C?
Without vitamin C, those hooks are never attached. The collagen strands just slip past each other. The resulting tissue is terribly weak, leading to the bleeding gums, loose teeth, and fragile blood vessels that we recognize as scurvy.
And vitamin D is similar, right? Without it, the machinery for calcium absorption fails, leading to rickets in kids and osteomalacia in adults.
Precisely. The machinery needs its coenzymes.
I really see the connections forming now. We also have nucleotides, which Oraby's text covers. Beyond just making up our DNA, free nucleotides act as the cellular power grid and communication network.
Right. ATP adenosine triphosphate is the power grid. Breaking the chemical bond of its third phosphate group releases the physical energy required to run all those active enzymes.
And then there's KMP, right?
Yes, cyclic AMP. This nucleotide acts as an intracellular messenger. When a hormone like adrenaline binds to the outside of a cell membrane, it triggers the production of SAMP inside the cell. That SAMP then cascades through the cytoplasm, flipping on the enzymes needed to meet the sudden demand for energy.
Okay, so we have the machinery, the materials, and the power. Now we run into a logistical barrier. How do we physically get the nutrients from our dinner plate into the cells while keeping toxic threats out?
It's all about transportation across borders. It begins with the violent chemistry of digestion, breaking massive structures down into molecules small enough to be absorbed.
Right.
In the stomach, the enzyme pepsin is tasked with chopping up complex proteins, but pepsin is completely inactive until it is exposed to hydrochloric acid.
And the pH in the stomach drops to around 1.2. I mean, that is remarkably close to battery acid.
It is, but the extreme acidity is necessary. It serves a dual purpose, it kills invading microbes, and it forcibly denatures dietary proteins. It unfolds those complex origami shapes, so pepsin actually has access to the internal peptide bonds.
That makes sense. But moving into the small intestine, lipids pose a completely different mechanical problem. Our digestive enzymes operate in water, And lipids are hydrophobic.
Right, oil and water don't mix.
So the fat just clumps together into large, indigestible globs.
This is where the liver steps in by secreting bile salts. Bile salts are amphipathic, just like the phospholipids we discussed. They act chemically identically to dish soap on a greasy pan.
Okay, I love that.
The hydrophobic tails stick into the fat globule, and the hydrophilic heads interact with the water.
Pulling the giant fat glob apart into millions of tiny microscopic droplets. Emulsification.
Yes, emulsification. It drastically increases the surface area, which finally allows pancreatic enzymes, specifically phospholipids, to access and digest the fat molecules.
Okay, so once broken down, these nutrients must cross the cell membrane, which the source describes as the fluid mosaic model.
Yeah, picture a sea of phospholipid bilayers studded with various protein channels floating in it, and it is selectively permeable.
Meaning only certain things get through.
Right. Small, uncharged molecules like oxygen, they just drift right through the lipid sea via simple diffusion, naturally moving from areas of high concentration to low concentration.
But what about bigger stuff?
Ions, or molecules moving against their concentration gradient, require active transport. The sodium-potassium pump is a classic example. It physically grabs sodium ions inside the cell, uses the energy from ATP to forcefully change its own shape, and pushes the sodium out while pulling potassium in.
And that electrical gradient is what allows your nerves to fire.
Exactly.
That brings up a critical question, though. The body builds these intricate gates to pull the good stuff in. How does it handle the bad stuff? Like foreign chemicals, toxins, prescription drugs, what the text calls xenobiotics.
Xenobiotics are any foreign compounds not naturally expected to be in the organism. And the problem is, most of these toxins are highly fat-soluble, meaning they easily slip past our cell membranes and accumulate in our tissues.
Which is really bad.
Very bad. So the liver functions as a two-phase chemical defense system to force these toxins back out of the body.
It's essentially a biological waste management facility.
It really is. In phase 1 metabolism, the liver utilizes a massive family of enzymes called cytochrome P450. Their primary job is hydroxylation. They forcibly add an oxygen and hydrogen group onto the fat-soluble toxin.
Making the trash chemically reactive, it basically gives it a handle.
Yes, a highly reactive handle. Then, in phase 2, conjugation takes over. The liver grabs that handle and attaches a massive, heavy, water-soluble molecule to it, like glucuronic acid, sulfate, or glutathione.
Okay, so it tags it.
Right. The physical chemistry of the toxin is completely altered. It is now highly water-soluble, meaning the blood can easily sweep it to the kidneys to be filtered out in urine.
So what does this all mean for someone taking multiple prescription drugs, like an older patient on 5 different meds?
This raises a hugely important question about competitive inhibition on a systemic scale. Many prescription drugs utilize the exact same specific cytochrome P450 enzyme for their phase 1 metabolism. Yeah. If a patient takes drug A and drug B simultaneously, and both need the same enzyme to be cleared, the system bottlenecks. Drug A physically occupies the enzymes, preventing drug B from being processed.
Oh, wow.
The clearance rate drops, and drug B builds up in the bloodstream to highly toxic, potentially lethal levels. This biochemical traffic jam is the fundamental reason pharmacists tirelessly check for drug interactions.
That is terrifying, but it makes so much sense.
Yeah.
Okay, we've traced the atomic structure, the machinery, and the borders, but we really must zoom out. When we view the whole patient, what happens when this entire biochemical supply chain breaks down?
We see clinical malnutrition. An essential metric here is nitrogen balance, which tells us if the structural scaffolding is being built or demolished. Dietary protein is our primary source of nitrogen.
Right. So positive nitrogen balance means intake exceeds loss. The body is hoarding building materials.
This is an anabolic state. We see positive nitrogen balance in growing infants, pregnant women, or athletes synthesizing new muscle tissue.
And the opposite.
Conversely, a negative nitrogen balance means the body is excreting more nitrogen than it is consuming. In starvation, advanced diabetes, or cancer cachexia, the body actually begins tearing down its own skeletal muscle just to harvest the amino acids for vital organ function.
The source outlines 2 specific, really extreme forms of protein-calorie malnutrition: marasmus and kwashiorkor.
Yes. In marasmus, the patient suffers a total deficiency of both calories and protein. The body enters severe catabolism, consuming all its fat reserves and muscle tissue. The clinical presentation is profound emaciation and muscle wasting. You can visibly see they are grossly underweight.
But kwashiorkor presents differently. This occurs when caloric intake might be somewhat adequate, perhaps through a diet consisting solely of starchy carbohydrates,
Mm,
But protein intake is virtually zero.
right.
And photos of these patients often show severe swelling in the abdomen and extremities, along with an enlarged fatty liver and skin pigmentation, which feels deeply counterintuitive. Why does starvation lead to swelling?
Well, if we connect this to the bigger picture, the swelling edema is driven by physical chemistry, specifically osmotic pressure. In a healthy individual, the liver synthesizes massive amounts of plasma proteins, primarily albumin, and pumps them into the bloodstream.
Okay.
These proteins are large colloids. They cannot pass through the blood vessel walls.
So they act like a sponge holding water inside the circulatory system.
Exactly. Water naturally wants to flow toward areas with a higher concentration of solutes to equalize the balance. Albumin provides that concentrated osmotic pull, keeping the fluid volume of the blood stable.
But in kwashiorkor,
In kwashiorkor, the liver is starved of amino acids. It simply halts albumin production. The sponge disappears.
and without the osmotic pull, the fluid just seeps out of the blood vessels and pools into the tissues. A massive systemic failure caused by a lack of raw materials.
It is a devastating cascade. And, these principles of physical chemistry, osmosis, colloids, and crystalloids are directly harnessed in modern medical treatments, too, like dialysis.
Right, a dialysis machine essentially acts as an artificial kidney, utilizing a semi-permeable membrane.
Yes. The toxic waste products in the blood, like urea, are small crystalloids. The vital plasma proteins are large colloids. The dialysis membrane has pores precisely sized to allow the small toxic crystalloids to diffuse out into the cleaning fluid, while the large vital colloidal proteins are blocked and safely returned to the patient. It is purely mechanical filtration.
It's incredible how we've weaponized basic chemistry to save lives. And speaking of radioactive isotopes, the source also mentioned using iodine-131 to diagnose thyroid diseases, which is just another example of physical chemistry in medicine.
Exactly. Now, before we wrap up, let's actually employ a clinical review exercise based on the mechanisms we've discussed.
Yep.
So, for you listening, consider a patient who presents with severe unexplained joint pain, bleeding gums, and a dietary history indicating literally zero consumption of fresh produce for several months.
Okay.
What specific biochemical mechanism is failing, and what is the missing component?
Oh, let me jump in here. Let me trace the logic backward. The dietary history of zero fresh produce points strongly to a vitamin deficiency, right?
Good.
The clinical symptoms, bleeding gums, and joint pain, indicate that the structural scaffolding of the blood vessels and connective tissues is weak. That means the collagen is defective.
Yes.
And we established that collagen requires its proline and lysine amino acids to be hydroxylated so they can cross-link. So the missing component is vitamin C, the necessary coenzyme for that hydroxylation. A clinical diagnosis is scurvy.
Flawless mechanical deduction. The dietary absence removes the coenzyme, the enzyme halts, the chemical crosslinks fail, the structural protein unravels, and the patient bleeds.
It is an incredibly clear chain of cause and effect once you actually understand the pieces.
It is. And I want to leave you with a final thought to mull over on your own. Every second, right now, as you process these words, millions of xenobiotic toxins are being actively tagged with glucuronic acid in your liver.
Mind-blowing.
Billions of competitive inhibitors are battling for active sites. Trillions of sodium-potassium pumps are physically altering their shape to push ions across your membranes. Your conscious mind is entirely unaware of this massive, synchronized industrial complex.
Yeah.
Consider how fragile this system is. A single atomic substitution, like replacing one hydroxyl group with a fluorine atom in your teeth's enamel to prevent decay, or shifting a single amino acid in a hemoglobin chain that can dictate the entire trajectory of your health. You are a walking, breathing, astonishingly resilient chemical machine.
Wow. Thank you so much for joining us on this exploration of Oraby's biochemistry. The more you understand the invisible molecular mechanics operating beneath the surface, the better equipped you are to understand the clinical realities of your own health. Until next time, keep investigating the incredible machinery within you.